Thank you very much. I'm very excited to present to you today my project, which is the controlled myogenic differentiation of mammalian cell lines by temperature-inducible systems. So, as we know, meat is one of the main components of meat is muscle tissue, and the process to form muscle tissue is called myogenesis. The myogenesis starts from cells called myoblasts that proliferate, and then they withdraw the cell cycle and they commit to differentiation, becoming myocytes. The myocytes then fuse to form myotubes that mature into the myofiber, which is the main component of the muscle tissue. Every stage of the differentiation is characterized by the expression of specific markers, like MyoD, ME5 for the myoblasts, MRF4 and myogenin for the myocytes, myosin heavy chain, creatine kinase, and alpha-actin for the myotubes. So we decided, MyoD is one of the main master genes that regulates the myogenesis. So we decided to create a cellular system in which MyoD can be triggered by an external biophysical stimuli to create myogenic progenitors and muscle tissue for cultured meat production. So among the all biophysical stimuli, we selected the temperature. So the idea is to expand the cells at 37 degrees, induce the differentiation by an increase in temperature, and then obtain the differentiated muscle tissue. So I will give you a very brief overview about the heat shock response, which is a physiological response that happens in every cells after temperature increase. So among the heat shock, there are several transcription factors that activate in the cytoplasm, and they bind to the DNA, activating the transcription of the heat shock proteins, which are a family of proteins that activates a series of pathways that are involved in cellular protection. And I want to focus your attention on one specific transcription factor, which is the heat shock factor 1. Heat shock factor 1 is present in the cytoplasm in a monomeric form, and upon the heat shock, it trimerized and it migrates to the nucleus, and it binds to specific sequences that are the heat shock elements that are present in the promoter of the heat shock proteins. So based on this knowledge, we decided to create two different vectors. The first one containing the HSP70 promoter with a multimerization of the heat shock elements to enhance the activation of the promoter. And downstream the promoter, we added MyoD, which is one of the master genes of myogenesis, and SMARCD3, which is a chromatin remodeler that is known to interact with MyoD to facilitate its binding to the target genes inside the nucleus. The second vectors contain HSF1, which, as I said, is the transcription factors that activates the transcription of HSP promoter, downstream CAG promoter. With these two vectors, we created two myoblast cell lines. The first one is the Isccm cell line containing only the MyoD SMARCD3 vector, and the second one, the Isccmh cell lines containing both the vectors. These cells went through an expansion phase at 37 degrees, and the differentiation was induced with an heat shock at 43 degrees for 2 hours and a switch to the differentiation medium. And then the samples were analyzed by real-time qPCR and immunofluorescence. So briefly, we seeded the cells, we induced the differentiation by heat shock and medium switch, and the samples were collected after 24, 48, and 72 hours. So to assess the efficiency of the differentiation, we went to analyze two different markers, which are myogenin and myosin heavy chain. For myogenin, we saw that the Isccm cell lines start to express myogenin after 1 day of differentiation, especially upon heat shock. But the expression of myogenin is significantly higher when also HSF1 is present. So we concluded that HSF1 promotes the formation of the myocytes. Regarding myosin heavy chain, which is the marker of the myotubes, we saw that we do have an induction in the Isccm cell lines upon heat shock, but this is particularly evident for the Isccmh cell lines upon heat shock. So we concluded that HSF1 in combination with the heat shock promotes the fusion of the myocytes into the myotubes. And we confirmed this by analyzing the protein levels. So we performed an immunofluorescence for myosin heavy chain, and we used the L6 cells as control, where we saw that after 2 days of differentiation, we have a very few numbers of myofibers, while for the experimental condition, at 37 degrees, we saw no differentiation at all, while upon heat shock, we saw that the L6MH cell lines show an increase in the immunoreactivity for myosin heavy chain, suggesting that we do have the formation of myotubes. These analyses were also confirmed by quantitative analysis. So we went to investigate the fusion index, which is the percentage of nuclei which is inside the myotubes. And we saw that in Isccm cells, after 1 day of differentiation, we do not have myotubes, while they start to form after 2 days. But in the L6MH cell lines, upon heat shock, we do have a high increase in the fusion index, which is statistically significant when compared to the other conditions. And the same we can observe for the myotubes area, which is the area of the myotubes out of the total area of the analyzed field. And we saw an increase in the area starting from day 1, and a significant increase through the 3 days time course. So we also wanted to investigate the induction of the transgenes upon the heat shock. And we analyzed the levels of MyoD and SMART-CD3. So here we have the induction of 43 versus 37 degrees. And we saw that after 24 hours from induction, we do have an increase of 3.5 times of MyoD in both L6M and L6MH cells, and then it decreases at 48 hours. And a similar pattern we observe for SMART-CD3, even if the levels of induction are lower. But the problem is that we saw no differences between the L6M and the L6MH cell lines. So as the heat shock is a very fast process, we try to investigate the induction of the transgenes at earlier time points. So we analyzed 4, 8, and 12 hours after the differentiation induction, and we saw that after 4 hours we have a peak of the induction which goes to 300 to 400 times higher after the heat shock, and then it progressively decreased through time, both for MyoD and SMART-CD3. But still, we did not observe a difference between the two cell lines. Therefore, we went to investigate the protein levels, and we performed an immunofluorescence for MyoD after 8 hours of induction. And we saw that at 37 degrees, we have a very low signal for MyoD, while upon the heat shock, we saw for L6M cells an increase in the signal and an accumulation of MyoD signal in the nuclei. And this is particularly evident for the L6MH cell lines, where the signal is higher and most of the cells accumulate MyoD in the nuclei. And this was also confirmed by quantitative analysis. So we went to investigate the intensity of both MyoD and SMART-CD3 signal, and we saw that for MyoD we have a significant increase in L6MH cell lines upon heat shock when compared to the other condition through the 24-hour time course. And a similar pattern was observed for SMART-CD3, especially after 12 hours of induction. For the accumulation of the signal inside the nuclei, we saw that for MyoD, we do have a significant increase in the nuclear signal upon heat shock, which is also higher than the L6M condition. And the same we observe for SMART-CD3, especially after 8 hours of induction. So from this, we concluded that HSF1 promotes both the induction and the nuclear accumulation of the transgenes upon heat shock. So in conclusion, we can say that myogenesis can be efficiently induced by a change in the temperature without involving chemical substances, that the myotube formation can be accelerated from 4 to 2 days, and that HSF1 enhances the differentiation process. Oops, sorry. Okay. As future perspectives, we want to investigate the protein interaction between MyoD SMART-CD3 and HSF1 to test different cell lines for origin, species, and myogenic potential, and also to move to an industrial scale for human consumption or pet food production. I don't know. Okay. Saying that, I want to mention that we have a pending application, pending patent application for this system, and I want to thank the Laboratory of Cultured Meat at University of Trento, the facilities at the university, and Bruno Cell, and I want to thank you all for your attention.
Thank you very much. Really great talking. Is there a question? Oh, yeah. You?
Thank you for your great presentation. The 43 degrees Celsius reaction for 2 hours is very interesting. Mm-hmm. Is it possible to transport the same heat to cells, all cells, in such a like a short time?
Well, it is possible to use a shorter time for the heat induction, but from the literature we saw that 2 hours is one of the most used time point for the induction, because it is the necessary time for the process to start.
Thank you.
Thank you.
Maybe one online question, and then we move on to the next talk. Oh.
Hiya. So we have a question from online about the future perspectives for 3D and suspension-based culture with these cells. And as kind of a follow-up, I'm also wondering, how do you envision this heat shock functioning in a large-scale bioreactor system?
Well, in the bioreactor, the temperature can be fine-controlled. So if you increase the temperature inside the bioreactors, it is possible to induce the system, the activation of the system, just by increasing the temperature in all the bioreactor. Maybe with some shears that can move the medium in order to let all the cells have the same temperature.
So thank you very much. I'm afraid we have to move on to the next session.